Staining techniques are essential tools in microbiology for visualizing and identifying bacteria. These techniques involve applying dyes to bacterial cells, which enhance their contrast against the background, making them more visible under a microscope. Common staining techniques. Gram stain. This is the most widely used staining technique. It differentiates bacteria into two major groups. Gram-positive bacteria. These bacteria retain the crystal violet stain and appear purple under the microscope. Gram-negative bacteria. These bacteria do not retain the crystal violet stain and appear pink or red after counterstaining with safranin. Acid-Fast stain. This technique is used to identify bacteria with a waxy cell wall, such as Mycobacterium tuberculosis. These bacteria resist decolorization with acid alcohol and retain the primary stain, carbolfuchsin. Endospore stain. This technique is used to detect the presence of endospores, which are dormant structures formed by some bacteria. Endospores are resistant to staining, but they can be stained with malachite green. Capsule stain. This technique is used to visualize the presence of a capsule, a protective layer surrounding some bacteria. Capsules appear as clear halos around the bacterial cells. Flagella stain. This technique is used to visualize bacterial flagella, which are hair-like structures that enable bacteria to move. Other staining techniques. Simple stain. This technique uses a single dye to stain all bacteria the same color, making them more visible. Negative stain. This technique stains the background around the bacteria, leaving the bacteria unstained and appearing as clear spaces. Importance of staining techniques. Visualization. Staining makes bacteria more visible under a microscope, allowing for easier observation and identification. Classification. Staining techniques can help classify bacteria based on their cell wall structure, presence of endospores, and other characteristics. Diagnosis. Staining techniques are used in clinical microbiology to diagnose bacterial infections. By understanding the principles of staining techniques, microbiologists can effectively visualize and identify bacteria, leading to better diagnosis and treatment of infectious diseases.